Hi. 11s and 12s. Sorry, just moved the camera a bit there. Hi 11s and 12s. What we're looking at here is a digital microscope. I will put a picture of this in the end to show the setup that I'm using. It is important when I'm going through this, we're doing this for online learning's sake. With the digital microscope, they have slightly higher magnification than a normal microscope would have that you use in the classroom. So for example, while I'm on the 4x lens here, with the microscope you'd use in class, it's 40 times magnification. It's quite significantly more magnified on this form. So worth to know that the calculations I do on this video will not be the same as the calculations you do if you're doing it in person in class with a microscope at home. But what we're doing here, it's not a slide or it's not a cell we're looking at or anything like that at the moment. What we're actually looking at is we're looking at a mini grid. So on this mini grid, what we're looking at here is we've got these little small squares here, and we've got the large squares. Now the small squares are equal to 0.1 of a millimeter, and if you add them all up, you get 1 millimeter, which tells me that one large square is 1 millimeter. So that tells me if I'm looking across this entire length, that I'll just move the microscope over a little bit so we can see some whole squares, start in the whole square. So roughly, I can definitely fit 2 whole squares across the slide here, and there's a little bit more. The best way of doing that, I suppose, it would be to get, if we go, try and get, so we know it fits 2 whole squares in. There we go. So we've got our 2 whole squares, so we know up to here is 2 millimeters, and then there's another 2 small, which is 0.1s. So over here, the whole field of view for this 4 times magnification, the objective lens, is going to be 2.2 millimeters long. So anything that I look at in here, if it took up the whole slide, is going to be 2.2 millimeters, and that helps set that scale. Now it is worth mentioning why we've worked out the diameter of this field of view is 2.2 millimeters. That's not useful when we're doing a microscope drawing. When you're doing a microscope drawing, you probably want to put your scale in micrometers. Now to do that, 1 millimeter is equal to 1,000 micrometers. So over here we can see, because it's 2.2 millimeters, the field of view for this would be 2,200 micrometers, and that would be what I'm using for my scale. So, now that we know that the field of view is 2,200 micrometers, what we're going to do is we're going to change the slide. So I'll just pause the video here, and I'm going to change the slide, and we're going to look at a cell. So what we're looking at here is we've got some striated muscle. So now that we've changed the slides, we're on the mini grid, now we're back on the striated muscle. Looking at the striated muscle, we can see, having a look over here, that the length of this kind of takes up almost 1/3 of the length of our screen. Knowing that it's folded across, we could say that's roughly 2/3 of the screen. So what we need to do is, knowing that the entire field of view is 2,200 micrometers, divide that by 3, times that by 2, and we could probably say that the length of this fibre here is roughly 1,466 micrometers, or 1.4 millimeters, 1.5 millimeters. So that's the way that we calculate scale for these. Now, drawing this, which is what I'm going to actually ask you to do, this exact picture, I'd like you to draw in your books using the drawing techniques in the video that we had previously released, is you need to be drawing what you see. So we know there's a whole lot of cells in here, but we can't see them. Okay, so what you're drawing is you're just drawing all these fibres, you're annotating it as much as possible, you're putting a scale, you're putting a title. So my title might be striated muscle fibres. I've got my scale where I would have to, I know that this itself, or I know that the whole length of this is 2,200 micrometers. Maybe if I draw 1 centimeter along of that, divided by how many centimeters would be in the total length of my drawing, and the scale will be that equivalent down here. Then you label what you see. So maybe tangled muscle fibres, dyed blue, things like that, or some dots over here, or you try and be scientific if you know what you're looking at. But that would be how you label a video, image such as this.